रिमोट है। हम्म।
वो माइक का है।
हेलो। 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 वेलकम वेलकम। हाँ। बात करने का टाइम नजदीक। हाँ, तो वही टेस्ट किया ना हमने। हेलो हेलो हेलो। माइक टेस्टिंग। माइक टेस्टिंग। हेलो। 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 हेलो हेलो। माइक टेस्टिंग। अभी चल रहा है। कोई बात है, इसमें क्या है। 8.8502. इधर इधर से कोई प्रॉब्लम हाँ तो कोई प्रॉब्लम हाँ ठीक
हेलो। वहाँ का तो चल नहीं रहा। हाँ।
Hello, sir.
हेलो।
Good morning, sir.
गुड मॉर्निंग।
Good morning, sir. Thank you very much for gracing this occasion. Sir, we are just going to start the function. Our guests are coming.
Yeah, unfortunately, I have to leave in the next 10-15 minutes. So you will have to be a little bit like, see if it is possible. Otherwise, I have to take it from the car, which will not be very good.
Sure, sir. Thank you. Thank you very much.
5-10 मिनट 5 मिनट हैं अभी।
Sure, sir. I will start. We'll start with the TG, sir. Okay. Thank you very much. Please have the seat. A very good morning to esteemed guests, distinguished members of the ICMR, faculty, researchers, and all those gathered here physically as well as online. It's with great honor and pleasure to welcome you to the second Foundation Day celebrations of ICMR National Animal Resource Facility for Biomedical Research. As we gather to commemorate this significant milestone, let us reflect on the remarkable journey of the NARFBR and celebrate its unwavering commitment to advancing biomedical research in our nation and beyond. To begin the program, may I kindly request our esteemed director, Professor Mukesh Kumar Gupta, Honorable Guest of Honor, Dr. Vikas Vohra, sir, our founding director, Dr. P. Suresh, and our former director, Dr. H.G. Ramachandra, to grace us with your presence on the dais. Please, sir. Okay, as we have our DG sir has some urgent meetings, so I would like to request our DG sir to address the gathering. Please, sir.
Thank you very much, and sorry to hurry you a bit. Otherwise, I would be leaving for the meeting in NCI Jhajjar, and it would be more difficult to have a stable internet connection on the way. So, unfortunately, I will have to say what I have to say now, and then you can formally welcome the others, all the guests, etc. So I don't know who all are in the audience, but I'd like to say that I am very pleased to be here online on the second foundation day of the National Animal Resource Facility. And I would, if they are in the audience, I'd like to acknowledge Dr. Vikas Vora, Dr. P. Suresh, Dr. Ramachandra, as well as the current director, Dr. Mukesh Gupta. It gives me indeed great pleasure to talk to you today on this occasion of the second foundation day of NARFBR. It's an institute that I am very proud of. I think every person associated with ICMR, and rather any Indian, should be proud of this facility. It's a really well constructed, well thought through facility with which has the potential to be the hub of preclinical research in India. So when Honorable Minister inaugurated the facility at the end of 2022, since then we have great expectations from this institute. And last year, or earlier this year, I don't now remember exactly when, the new director, Dr. Mukesh Gupta, has taken over this facility. And it has already shown that it can work as a very important hub for preclinical research. In particular, I'd like to say that when we have opened what we call the MedTech Mitra, a portal through which all the innovators or any innovator, public or private, academic or research institute or industry, can come and ask for not only advice, but also help with many things from A to Z of innovation, from trying to develop the concept and making a prototype of a diagnostic, a device, or a candidate vaccine or a candidate therapeutic, right down to regulatory strategy and getting the preclinical research done, particularly in larger animals when necessary, clinical research, including phase 1 studies, but continuing to phase 2, 3 studies. And then not only that, but after approval of the CDSCO, cost effectiveness analysis, inclusion in guidelines, and taking to market, both in terms of procurement by the public health system, as well as use in the private sector. All these steps are part of MedTech Mitra. And the National Animal Resource Facility is an important partner in this whole process because it fulfills that preclinical research window. But we have to go a very long way. We have to get GLP certifications for our labs. We have to make sure that any type of experiment preclinical study is possible to be conducted in NARF. And in the heart of Genome Valley, this institution is and should become a center for interacting between academia, research institutions, and industry. So our hopes are very high, and I'm sure all the scientists working at NARF and the new director are working extremely hard to make those expectations a reality. I again welcome you all to this Foundation Day and give my best wishes to this institution, which I'm sure will grow to be one of the best institutions, not only in the country, but in the whole world when it comes to preclinical research. Thank you. Thank you all very much.
Thank you very, very much, sir, for giving your valuable time for your address and inspiring us towards a brighter future in the biomedical research. Your profound experience and leadership in the field of medical research has been instrumental in guiding our nation's healthcare landscape. Thank you very, very much, sir. Now I would like to request our guest and guest of honor director, our former director and our founding director to occupy the dais and grace the occasion. Thank you very, very much, sir. नहीं, okay, okay, ठीक है. Now I would like to request our Honorable Director, Professor Mukesh Kumar Gupta, to present his welcome address and share his invaluable insights with us on this momentous occasion of our Foundation Day celebration. Please.
Dr. Rajiv Bahal, Secretary DHR and Director General of ICMR, our Director General, I would rather say. Our invited guest, Dr. Vikash Bohra from the NDRI Karnal, our founder, Director in charge, Sri P. Suresh, Dr. S.G. Ramachandra, our former director, other directors who have joined online, people from the CPWD, ADG, my colleagues, my staff members, all the people, a very warm and hearty welcome for the day. Today is a very, very important day for all of us, as we all know, that the foundation of this day was laid way back on 2018 on this particular day, that is the 7th of May. And this institution was inaugurated in 2021, December. It was inaugurated. And today we have gathered here to celebrate the fruits that we are having from the foundation that was laid on 2018. Today is the day where we also take opportunity to pay respect to the people who have been behind the establishment of this institute, which starts with the ICMR Director General, the secretaries, and many other people who have been behind the backbone of this institution. I take this opportunity before our Director General leaves this meeting to thank you very much for all along their supports. Since I joined this institution, he has been supporting us with his wisdom and a lot of guidance that he has given to us. how to take forward this institution in becoming an institute, a real institute that should become an indispensable for the preclinical research in the country, and to how to be a key partner for the MedTech Mitra and other programs of national importance of the country. As we stand today, we have the resources, livestock resources, laboratory animal resources for the almost all major diseases that we can expect to be have the potential of becoming a pandemic or even the epidemic in the region. I'm very happy to share with all the people that we have more than 11 different types of transgenic animals with us, which are very vital for doing both the basic research, development and testing of vaccines, biologicals, and other the pathogenesis of the infectious diseases having the potential to become a pandemic. We also have the other laboratory animals like hamsters, which we acquired recently. Then, of course, the guinea pig, rabbits, and large animals. I'm very happy to share that to our DG sir and other the invited guests, that now we are our country's first institution to have the lab-grown dogs, beagle dogs. We are the first one in the country. We have lab-grown dogs now we have lab-grown monkeys we had, and now we have a good population of that, non-human primates. We also have the lab-grown pigs now we have for our study. We also have our lab-grown goats with us. So I'm emphasizing the word lab-grown, 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 because these animals you can find throughout the country. How, and where we differ from the other is that these are grown in-house. These are not the animals now which are meant for eating purposes as a food animal or as a pet animals. Dogs are pet animals. The pigs and goats have been used as the food animals. But what we have is no more a food animal. They have been housed, reared, monitored for the genetic makeup and health as per the regulator's requirement and in a uniform conditions, so that when you perform any kind of experimentation, any kind of preclinical trial, they give near uniform results. They have been genetically monitored that they do not have any genetic instability, and they are also monitored for their health status, including to ensure that they are free of specific pathogens as required by the regulators. When you take an animal from the say, for example, dogs, which people have been taking, capturing it from the breeders, taking it from the breeders, or taking the monkeys, capturing from the wild, they have been grown in diverse environmental conditions, exposed to the different pathogens or other microorganisms, have the different status of the immunoglobulin status within them, and they are a great source of variability within them. What we have is entirely different from it. They are grown in a ISO level 8 facility in our condition, where they are not exposed to the pathogens. They're very less particulate matters, as we all know it, 100,000 particle, less than 100,000 particle per cubic feet. A nicely constructed facility by CPWD, we acknowledge that. And these are monitored at our facility routinely. So these laboratory animal is something which we take proud of. And we have now almost started taking strides to get ourselves registered and certified as a GLP facility. GLP requires certain experiments to be performed before we apply for the certification. As we all know, by this time, we have already completed a few trials and we are on the go for the GLP certifications that is in the pipeline. But besides that, I'm also very happy to share that we have also been working on the alternatives to the animal models as one of the goals toward the 5R. Earlier, we used to talk of 3R. Now we are at the stage of 5R. And we have now the secondary iPSCs, induced pluripotent stem cells. With us, we can generate them. We have the potential to do that and develop organoids, which we will be able to supply on demand to any institutions who want to work on the secondary iPSCs, and they are organized for drug testing or testing of the other biologicals or pharmaceutical products. Along with that, we also have the many activities in the pipeline. We have several services we are offering now to many, both government and non-government commercial companies like biological events or the Santa Bio or many other pharmaceuticals, biotech vaccine companies we are offering. We are also trying to popularize goat, which India has more than 25% of the goat population in the country. We are trying to explore and popularize how we can make goat as a unique model for human diseases such as osteoporosis. We also is one of our focus area for this institution. We are also health monitoring within the laboratory animals, and we are also developing and trying to develop countries first, probably countries first, miniature swine for biomedical research. Hopefully next year when I'm talking to you, we'll be having the country's first miniature swine for biomedical research with us. So today's day, I just wanted to reiterate where we are heading, but then also to remind us ourselves that the foundation for whatever we are today was laid down way back to 2018, and 2 years we have come to a certain stage where we can say that we have actually moved an inch. And for this, I'm happy to see that the founders of these institutions are with us. We have Dr. P. Suresh, who has been the initial stages, the backbone for development of this organization. What S.G. Ramachandra, our former director, who is also with us, and I'm very thankful for them to grace this occasion and then to give us further direction with us. And we'll be very lucky today to have the lecture from Dr. Vikash Bohra, who knows, I think everybody is working in the field of genetics and genomics of livestock, how to get them, identify them, how to breed them in a systematic manner, and how to get them registered and identify the new breeds or new strains or the new species. He is the champion of that, and we'll have the opportunity to meet with them. He needs no introduction for all those who are working in the field of the identification of the newest species or newest strains of the. He has identified several desi cattle and other livestock in our country, either directly as a PI or indirectly as his OPI, but he is the one which has. So once again, welcome you all for coming to this occasion, and I look forward to having a wonderful interaction among all of us. And before I close, I also request all of you to please either switch off the mobile or put them in vibration mode so that it does not distract others. Thank you very much.
Thank you very much, sir. Your words of wisdom and guidance will truly illuminate and inspire us all towards greater achievements in the field of biomedical research. With great pleasure and honor, I extend a heartfelt invitation to our esteemed guest of honor, Dr. Vikas Vora, sir, who is the head of the Division of Animal Genetics and Breeding at ICMR-National, Karnal, to address the gathering. Please, sir.
Good morning. And first of all, happy Foundations Day to all the staff, to my good wishes, Honorable DG, DMR, DHMR, and DG ICR, ICMAR, MR, sorry. So Dr. Rajiv Bahl, sir, he was there online few moments ago. Honorable ADG, Dr. Bansal, my friend and colleague, Dr. Mukesh Gupta, and Dr. P. Suresh, Dr. S.G. Ramachandran, the former founders. In fact, Dr. Mukesh will also be considered this as a founder member. So, being a man of genetics, at the outset, I would like to share my one of the concepts of genetics, which is called as founder's effect. So I'll take the attention there. So for the house, those who don't know, founder's effect is the effect which can never be overcome in the subsequent generations. So all the founders, what I could come to know from Dr. Mukesh, Dr. Suresh, Dr. Ramachandran sir, whatever stones they have laid, of course, the blessings and the guidance of the seniors, the DG and the ADGs, their support is required. But I think largely the coming generations, Dr. Mukesh has to follow that leads and take it much more ahead. So then comes the next concept of genetic improvement, you know. So the improvement will be done by the Dr. Mukesh and many more to come. Definitely, this is a biomedical research institute, and I have a very little exposure, frankly speaking, of ICMR. But since now my friend is there, so I think I will learn from him also, though we are characterizing the large animals. But the importance of small animals and preclinical trials, which in a large animal we are lacking there. So I think this resource facility can serve as an excellent medium and a collaborator with us, that if we want to work something related to the humans, like our institute is also working on some prebiotics, postbiotics, and then comes the question that you need to have an accredited lab where you can validate your experiments, do basic validations in the animal models. So such facilities are really required and they are very limited. In ICR, I think to best of my knowledge, no such exclusive facility where animal genetics are being maintained for preclinical trials or drug testing are not there. So we often look at either CSIR laboratories or ICR and to work with them and see. So the importance of this institute is very, very huge. You know, Dr. Mukesh is trying to grow and breed the all species. He has a target of maintaining all preclinical what we say, the animals. So that all aspects are covered here. So looking into that, I wish him all the best. And this is the institute. I can see so many familiar faces. So as a scientist, so they will also give their sweat and blood to nurture this institute. And keep in mind that the founder's effect should not lead to it should lead to a positive and positive effects of this thing. So with these few, my best wishes. I thank Honorable Dr. Mukesh for giving me this opportunity to interact with you all, and we'll share on characterization and how to identify the breeds later today. Thank you very much. Thank you.
Thank you very much, sir, for your kind words. May I now request our founding director in charge, Dr. P. Suresh, sir, to address the gathering and share your valuable thoughts to inspire us. Please, sir.
Thank you. Respected members on the dais, and respected members of the dais, my colleagues, friends. It's indeed a very happy moment for me, especially because this is the place which I was dreaming more. And it was an emotional, this one also an attachment for me, because day in and day out, almost for last 15 years before we actually got the approval, I used to go to Delhi and plead almost every secretary, every DG, everybody to accept our proposal and, consider this as an institution which will definitely get the good name. I used to tell them that this is the institution which has a lot of scope is there, and in India, nobody is having this type of institution. And one day, I have, in frustration, because when we have submitted 21st October 2004, I have submitted the EFC. There was no response from the ministry. And 2006, of course, we were having some internal meetings in ICMR, and I was asked to give some lectures in presentations in the ministries and all that. In 2005, 2006, but there was no response. So in frustration, I have written a letter to the President of India. You believe me, at midnight to 2 o'clock, I wrote a 2-page, very frustrated letter. And on November the 16th November 2006, that was. And suddenly on 18th January 2007, I got a phone call from the ministry, the Rashtrapati Bhavan, saying that, sir, President wants to talk to you. Then I said, okay, how much time will I get? He said, 2 minutes. No, I don't want 2 minutes. I want 10 minutes. I want minimum 10 minutes, maximum whatever the plan. No, sir, you won't get that much time because he's a very busy person and there are so many other people are also. Then I put down the phone. That was a black phone, classical, our black phone. This one, I put down the phone. Then he rang up again. That don't put down the phone now. I will give you the last this one. I will not. I will give you your appointment will be the last this one, and you will talk as long as he permits you, like that. Then February 1st, he is coming to Hyderabad. You will talk to him, and you can. So in Raj Bhavan, we heard all the directors and other things. Everybody, the senior colleague, the present director, Dr. Hemlata, Dr. Shashikaran, our director, they all went there. But they have not permitted them. Only Dr. Suresh, because he is the one who wanted to talk to him, so nobody else. Then they all went back. So I was the one who spent time with him. Almost like 35 minutes to 40 minutes, I spoke to him and explained him. Then afterwards, 2007, 8th November, we got the in-principle approvals from Planning Commission. I told him that please take this copy of the EFC. Then he said, no, I will not take any paper. You give me a small slip. Then I gave in a small slip that this is an NARF, this one. This is in IFD, Integrated Finance Division in Planning Commission. So you please take care of this. So we got the approval. Then I felt that was a very happiest moment. And the second happiest moment was the EFC 9th May. May is a very nice month, actually. May is a very nice month, why I'm telling, because 3rd, 4th, we had an international conference on stem cells in Hyderabad, and DG ICMR came and we proposed this one, and secretaries were there. And 5th, secretary called up and said, please give the write-up, then we will approve this. Then 6th morning, I went to Secretariat and submitted the 2-page write-up justification for allocation of the land. And the 10th May, we got the fax message that 100 acres of land has been given to me. And they told for the establishment of this. Then I wrote a letter to the DG ICMR. He wrote on 17th, he replied that, go ahead with this one. Professor Ganguly was very much encouraging. He told wherever you want to go, you go. And in 2003, I went to United States for 1-year training. He said me, in 2002 itself, he told me, no, you go to CDC immediately. In next month itself, I went to CDC Atlanta, and Jonathan Richmond was the director, and I spent some time there. 10 days I was there only, and came back to India. and started writing for the longer duration fellowships to learn about the primate facility, because our aim was to establish a primate facility in India. There is no primate facility at that time. Small animal facilities were there plenty. So they were also interested in establishing it. So I was sent to National Primate Research Center, Seattle there, and I learned the techniques, whatever that is required for the breeding of the non-human primates there. Dave Anderson was my mentor and collaborator. Actually, I've been given only 5 minutes time, so I've written this one in minutes. I'm sorry, I've taken 5 minutes. I'll take to read whatever it was. It occurred to me in my mind yesterday night, so I thought I scribbled it. On this occasion, I would like to congratulate all the members of the institution who have joined recently to serve this institution. My hearty congratulations to and welcome to this organization. It's a great institution under ICMR, I'm telling you, because this is the institution which will get name directly, because when you do any research in the institution, we may be restricting ourselves and we may be doing for ourselves this, but this is an institution which will serve the biomedical research. Many biopharma, biotech, biomedical institution, medtech institutions and all that, they will be remembering you, and the moment they want to start any project, they'll be remembering NARF ko jaiye, they will give you the product. So I have been telling thanks to CPWD members, Sridhar, and members have come, Srinivas, and so I really thankful to this, and they have made it such a nice institution. It was standing fourth after the first one is the Parliament Bhavan, and it ranked fourth. Correct? No, it is ranked fourth in terms of construction. This one, and all specifications, whether it is a mistake or whether it is a praising this one, all specifications I am responsible. All the specifications I have given. Some of them have not been implemented. That's a different story. But all the specifications, inch by inch, have been given by me. I'm proud to say that. So I'm really happy to see, very, very happy to see this facility coming up to expectations of our dreams and all that. This was the day I was dreaming, actually. Those days when nothing was there in this land, there were people who came to beat me also, the locals. They were doing the stone cutters and stone people. They were taking the stone and the mud and all this from this institution. So what they were telling is, this man becomes, he will. So they tried to beat me also. Then my driver, they protected me and took away from me this place. And somebody put a cross also in the center of the land. Then I threw that cross. I'm sorry if anybody is there, but I just went off throwing that one outside this. So they wanted to beat me because, they wanted to occupy this land. So almost 15 years without, because in 2000, from 2002 to 2016, we kept that land intact like that only. So without this, and I was all alone. There is no other person assigned for this particular institution establishment. So I was the project coordinator. So at that time, we were not getting any approval. We were not knowing what to do with this land, because enormous delays were happening in the ministry. So much of correspondence, providing clarifications, etc. It has taken many years for getting the approvals in ministry departments. But every department used to question us and give us the clarifications. The state governments did not take much time. Unfortunately, state government with the two papers writing itself, I've been asked to give the justification and just wrote there in the secretariat itself and given it, and they allotted the land. This is a surprise. And my sincere thanks to Sri Acharya ji, and who is instrumental in it. Mr. Acharya is a very dynamic person. All the excitement began after that only. And there was delays in every step, in various stages of the ministry. And from our side, we did not lose the track sequence, and we were following all this. Whenever there is a paper or clarifications comes, I used to reply within no time, within minutes, and within this one. There is no delay from our side. Only delay is from the other side. So finally, the most excitement day has come, 9th May, and 2014, we got the EFCA approval. And from then onwards, we were sure of getting this institution. So from that day, it was proposed 2nd August to 13th August 2014, we were going round and round and pillar to post. And those things corrections and making modifications to the document. Always we were, and the original proposal of this institution was PPP mode. PPP, public-private partnership, because we were expecting the most critical review. I mean, what is required is the efficiency and timelines. Most important is the timelines, which will come only through private organization. Government institutions, they tend to have a relaxation in terms of timelines. Oh, karenge aisa bolke. But most important for all this one, see, timelines are very, very important for submission of the projects and reports. And that's why we wanted to put it in the PPP mode. But later on, when Bhavani Tyagarajan secretary came, so she said, no, it is a government institution. We'll make it, all those things we can implement it, not a problem. So she made it. And then I had to rewrite the entire document once again. Then finally, thanks to our Prime Minister and the Cabinet Committee that has given approval on 18th November 2015, and EFC meetings, the major questions was, what is the demand? What was the supply position in the country and availability of different models and drugs, vaccines, testing and research in government institutions and private organizations? What is the plan for meeting this demand, which is very, very important. I had to give a business document also, and we had given what are the inputs and what will be the outcome of this one. I told even recently also when I was writing for the kill tanks, which is a very small amount. Actually, it's a 4 crores. I told I have been pleading ICMR, sir, please give us kill tanks. This one, which is only just 4 crores. I can run with one experiment. When COVID time, somebody rang up and told me that you do one monkey experiment, I'll give you 4 crores. Like that, it's a very small thing. So, we have delayed almost like, I don't know whether it has come. Ramchandra must knowing whether kill tanks have come. We can earn 4 crores within no time, actually. I have a party which will give you with one experiment, we can get 4 crores. I told this ICMR several times, sir, please sanction. So what I had decided, every day I used to write letters. Every day from my office, one letter should go to ICMR. All pending items I used to. So, unko bore kar diya, bahut sataya maine bahut sataya. I'm telling you, DGs have sataya. So administrative, they don't like me now. They don't like me. Because every day I used to write letters. So my sincere thanks to all the Director Generals, ICMR, who have encouraged me right from the beginning, especially Professor Ganguly, sir, with whose encouragement and with this one only. And throughout this one, he has been encouraging me. And Dr. Katosh, sir, and myself, we were sitting in the Finance Ministry. I was making a presentation for EFC, and he was helping me so much. Dr. Soumya Swaminathan, she made me, because I was having only 2 years service was there. She told, if I advertise, some new director will come, but you worked so hard. I don't want any new director, so you have only 2 years services there, so I will make you as a director in charge with all powers, like that she told, and she has appointed me, Soumya Swaminathan, and Dr. Balram Bhargava, sir. Actually, this facility where you are sitting right now, this auditorium, Aditya knows. You remember in Delhi, I was telling, this is a auditorium is required, sir. Then we have written the estimate, 4.95 crores, if I remember correctly. 4.95 crores hota hai. Please sanction it. Okay, you meet from the existing amount, available amount. Okay. This floor we got exclusively after that. Then I said 2 rooms, VIP rooms, sona hai, either, because this is a training program. The international members will be coming. Somebody wants to go. They cannot come from the city. They have to come and stay here only. So I wanted 2 rooms for the VIP rooms. Then archiving room is there, classrooms there. This is an institution meant for imparting training. So I wanted a floor. He was very much angry. You remember, he always used to be very angry. So kuch to puchta hai, always, sir. So both sataya maine, and every day I used to write letters. Something from my side, there is no delay, sir. So there are many things that I need to. I've traveled almost 36 times in a year. And people used to ask me, Suresh, where are you? Are you in Delhi or in Hyderabad? The local people who want my help and all that. So I'm in Hyderabad only. Like this. Then they used to put their request. So I said, whenever there is a requirement is there, I used to go there. So one day I was having high fever, I must tell you, because 2 o'clock in the afternoon, they called me and said, tomorrow morning you have to be there because ministry is asking you to come. Ambumani Ramdas. What time I should be there? 7 o'clock. So I went by 3 o'clock flight and, went there in a private, whatever is this flight available, you just come there. high fever. I went there in the morning, and 7 o'clock, believe me, 7 o'clock to 12 o'clock I was there. Minister has not come. And he has given me time at 7:30, 8 o'clock or something like that. Thank you so much.
Thank you very much, sir, for your kind words and taking us through the journey of the NARFBR. May I kindly request now Dr. H.G. Ramachandra, our former director of NARFBR, to address the gathering. Please, sir.
Good morning to all of you. First of all, I would like to thank Dr. Mukesh for inviting me to be with you today on this auspicious day. Thank you, sir. And my dear colleagues and friends. It's indeed a great honor for me to be here today with you and share my thoughts also. I think you heard a lot from Dr. Suresh, how difficult it is to establish an institute. I think he struggled almost for 2 decades. So, and finally he could do something and establish the great institute. I think this you can very well emphasize by he is telling about the statistics and the date. See, he remembers so much because he has spent all his time for this institute. So thank you, sir. Thank you very much for your all. So I was only part of his dream. I came at the end of his career. So I could do something and whatever unfinished by him. So I tried to finish and a little bit whatever I could do, I did in 2 years. And now the legacy is in the hands of Dr. Mukesh. I think he'll take it to the next height. So I was discussing with him and he was telling me today morning that this institute is going to be the Southeast Asian hub for animal resources. So it's a really great initiative and we all should join hands with him because nowhere in Southeast Asia, entire Asia, not only Southeast Asia, we don't have this kind of big facility. So at least if he can achieve that one, certainly it will be a big boost for the country itself. Because now India is a preclinical hub. I don't know how many of you are aware or not. So India is conducting around 60,000 GLP studies in India every year, the biggest compared to any other country in the world. So we are generating around 8,000 crores every year only for preclinical testing, GLP preclinical testing. It's a huge amount what India is generating. And NARFBR also is going to be part of that because now Dr. Rajiv Behel was morning telling that it has to be GLP certified facility. So to create a GLP facility, it takes time. It's not that easy. The contribution from each and every one of you is very, very important. And so if it becomes a GLP certified facility, certainly no one can beat us, especially in the government. There are private companies that are there, but still in government setup, there's no huge infrastructure like this. And so I all request you to join hands with him to make it GLP certified facility because this is a quality system. So if you have the GLP system, the data generated in NARFBR will be accepted worldwide, around 96 countries. There are 38 OECD countries and 56 associated countries. So in all these countries, the data generated from here will be accepted without asking any single question. So that's the beauty of the GLP system. Please be a part of GLP system and then try to contribute to the country also. So this is from my side from the GLP perspective. I have one more small request to Dr. Vora also. Dr. Mukesh was mentioning that so he's going to start a miniature pig breeding center here. Right now, so we have a miniature pig in Kerala. It's called Ankamali. So that one drawback with that pig is it's black in color. Skin is black in color. But that is the FDA approved pig breed. They are using for biomedical device testing. But the problem is now recently, not only recently, the FDA has approved the miniature pig for skin testing, skin irritation and the dermal irritation testing. But the problem is black color will not be able to appreciate the skin reaction. So we need to have white skin. So if we can convert that black skin to white skin by using some advanced technology, so that will be of great help because each miniature pig will be costing around 4 lakh per pig I'm talking about. Whenever you are importing, it's a huge money. Suppose if you have breeding facility here, all the contact research facilities in the country can use this miniature pig. So that's one small request, whether you can use some technology to change the color of the skin. Rest all everything is fine, only the color. So that's one small request from my side. So yeah, so that's no because Yes, yes. Because how albino rats were created. No, it was originally from brown mouse only. So it's genetic technology they have used and they have bred the white color. So like that. So it's a small request. So in addition to that, I am seeing a lot of faces here. Family is growing. So it's good to see that. And wish you all the best. Thank you again.
Thank you very much, sir, for your kind words and inspiring us. Now, I think we would like to have one break. And after break, we are going to have one special lecture from our guest of honor. So there is arrangement of some snacks in the canteen. Uh, Here only? Okay. So please have the snacks and refreshments. ठीक है. Okay. Please join us for the snacks and refreshments. research projects and initiatives aimed at unraveling the complexities of animal genetics and breeding using whole genome sequencing. May I request you, sir, to please come and deliver the lecture. Please, sir.
Thank you. So before starting the talk regarding the breed identification, so my domain has been broadly the cattle and buffalo breeding. So my talk is largely focused on that, but let us see what we can discuss. Feel free anywhere in between to interact and discuss if something is need to be known. Initial hiccups, I think it is ready, but this is on. One click. So when we say animal genetic resources, so these are what we are: animal genetic resources. We have animal genetic resource facility in the name of this institute. But I see that this is a very limited, say, world that we can have an institute to limit the genetic resource what we have in livestock. So we say that animal genetic resources, broadly called ANGR, are those animal species that are used or may be used for food and agriculture, and includes the population within the species as well as their stored genetic material. So many of us won't understand that stored genetic material is also our resources. And ANGR are the most valuable and strategically an important asset a country has. And India is a well-known biodiversity hotspot. So we have nothing but the diversity. And as the slogan says, unity in diversity. So we have a diverse communities, we have diverse genetic resources, we have diverse species, we have, and these are the raw materials which we say that we can use it for the further improvement, a breeder's raw material. Yeah. So in one slide, if I would like to summarize the management of animal genetic resources. So first and foremost step is we must know what we have. So it is called as identification. We must know the worth what we have it with us. So we evaluate it and we must describe our resources. That means we characterize it is the third step. Either you do it by phenotype, genotype, combine, or any other approach. If we come to know what we have and we have characterized it, then we need to improve our resources by selective breeding, upgrading, crossbreeding. In large animal, we say progeny testing program and marker assisted selection also. If we have markers for certain diseases, certain production related, fertility related genes, then if we found out all those things, we must conserve our resources and we must prevent them from declining or becoming an endangered. So the final thing is what we is a buzzword, we say sustainable management of animal genetic resources. So it is nothing but the using them in a judicious manner so that we fulfill the need of present as well as we pass on these resources to the future generations so that their aspirations and needs are also met. So how to measure the diversity? It is generally available in terms of species, breeds, populations, lines, and strains. So the larger the animal, the largely it remains either at breed level or species level. We can't go beyond that. Populations, lines, and strains are possible in small animals, small ruminants, lab animals, primates, because they have a smaller generation interval and variation for one particular trait. Even if you have built up a poultry for egg laying capacity or with a hard shell of an egg so that it has a good transportation quality. So that also comes under a strain or a line. So this is largely where we see our cattle and buffaloes of India. So it is genus Bovis, Bos, that means a hollow horn. And there is another breed so called Taurine, sorry, Taurine breed. This is also one of the separate lineage for this, and all European cattle are of Taurine breeds. Simultaneously, there is an synchral Kaffir African buffaloes, and there is a separate lineage. So this is basically, I have given a snapshot for bovine and in buffaloes. So there are three types of buffaloes: water buffalo, or what we call as Bubalus depricornis or celebs of Indonesia, and Mindora of also known as Bubalus mindoris. So in water buffalo comes the our buffalo of India, what we have, and there are at present 20 breeds of it. So defining a breed itself is a very tough thing, and FAO has done it for us. But what FAO has done is has given a very elaborate definition. So let us look at it. So it is a subspecific group. Species means subspecies, species or specific group of a species with a definable and identifiable external characteristics. That means visual appraisal is required to appreciate that this is different from different. One population, one breed is different from the other one. Or similarly defined group within the same species. Or a group which is geographically, culturally, and phenotypically separated. What is phenotype? What we see and measure is a phenotype. So if you say that height of the animal or body length of the animal, so they are also a phenotype, and milk of an animal is also a phenotype which you can see and measure it. And this separations, either geographical, cultural, or phenotypical, has led to acceptance of its a separate identity, and or a group of geographical or a cultural separation from phenotypically similar group has led to the acceptance of separate identity. Beyond this, also we define breeds based on the communities, like Reka and Ravaris. They are rearing the camel breeds, and these breeds are linked with their social, cultural, religious things. In south here, down south, Nilgiri, and we have Toda buffalo. So that is also having some religious utility based on which the animals are being reared, and reared in the semi wild type of management. So there are other ways where we also define the breeds, but let us largely focus on the first and foremost thing of a breed is that we should visually able to say that yes, this is different from this. The next step is that we then found that whether this variation is at random or it is actually due to some cause. So then we go for the cause. So the population lines and strains, we say, so a population is a group of animal within a breed sharing some physical or biological similarity. That means unique traits, unique characters, what we are identifying. If that is also there, so we can say it as a population. So within breed, variation is there, and there is one population with this, like Nili Ravi buffalo is there. So it has 5 distinct white markings. So all the populations breed doesn't have those 5 markings. Some have 3, some have 2, some have full 5. So we can say it's a population of a Nili Ravi buffalo. Lines and strains are attributed in animal genetic resources to particularly pigs, poultry, and the smaller animals thereafter, which have large litter size, small generation interval. So you can have a line. Line means having a common descendant, and we can say this is the line which we have continuously maintained from that particular ancestor, which was having some unique characters, right? So you can develop a line. Largely, we say an inbred line. And strains are also there. In poultry, we call it sometimes strains because good character of a yellow color egg and a brown egg can be a different strain, but the breed could be same, right? So mating systems, if we come upon, so there are two types of mating system. One is when the closely related animals are mated or bred, that is called as inbreeding. And the other one is when they are not closely related. When we say closely related means that the average relationship among in the population, in the herd, your farm, those individuals which are related on an average more than compared to the others. And we say outbreeding means they are not having any relationship. This relationship is used further when we go for molecular markers as well as the whole genome markers. Then we need to take into account this uh, relationship matrix into an account so that a marker and its effects is known. So mating system is what we observe to do maintain a farm or breed. Outbreeding is simply that we want one breed is having a good character, second is having another good character. So in the third generation, next generation, we want a breed complementarity, that we want both these characters to come and stay in this line. So we say crossbreeding is being done. Sometimes some resources are there which are not so well defined, and we want them to be upgraded to some purebreds. So we do upgradation and some experiments for hybridizations also. Outcrossing is most widely used breeding system in most of the domestic animal species, and the animals which are not related with the same breeds are mated. So this is different, a little bit. We follow it in human beings also. When we go for the marriages in down south here, there may be some different thinking, but in north it is said that we do not cross the related lines, right? So they tend to leave some lines and then they can go through what we say. Huh? So what is a purebred animal? Purebred animal is a animal which is born from true to breed. What we are lacking in here, because in Western world, everywhere, the purebred animal need to have some document, some registry, some breed registration, some breed society, which is maintaining the records of sire and dam. Except horses and dogs, we are not able to maintain these pedigree records or herd books or stud books in largely in our country. So we have a system of meeting. But if we have to go for visual appraisal and we say that what is a breed, whether it is a pure breed or not. So you know that coat color is highly skin color. In animal, we say coat color because they have hairs color different and the skin color different. So what we say coat color is the actually the color of hair as well as the skin color, which gives us a reflection in what we see at the animal. So pure breeding, if we say coat color is highly variable. So purebred animal need to have all the progeny of almost the same color. If we have male, female of this color, so all are having almost same color. So without saying anything, visually we can say, yes, these are. Definitely we will look for other characters that this breeds has. Sorry, it is not moving there. Oh yeah. So largely like these characters like this loose skin, the presence or absence of hump, the size of hump, the forehead, the orientation of the ears and the teats and udders. These are the traits. I will come to that later, but let us see if this is a purebred animal. So the skin color is almost the same. Then crossbreeding means when we are crossing for to gain something. So broadly, we are using a male, and male is an exotic breed, which is not a breed of our country. And we mate our females with them to produce a crossbred so that we can have good traits of male as well as females together in the generation. But what happens in the crossbreeding? So this is a famous example where we have built a Frieswald breed, and this is done by crossbreeding. That pure Holstein Friesian has been crossed with pure Sahiwal, and you see the difference in color. Variation in color is from reddish to black and white with equal surface area or patches, you can say. So this type of variation is created in the generation. And this variation then to breed further subsequent becomes a challenge. Because gene segregation, what Mendel told 200 years ago, is still relevant, and there is no rebuttals, no questions asked to what Mendel has said. So it do segregate, and which genes are segregating, we can't direct them. Okay, that will happen by nature. Something like called outcrossing. So there are two examples in this pictures that we are crossing the individuals which are not related, but of the same breed. So they also have the same phenotypic appearance, or visually, physically, we can see that they are the same. So the concept of grading up is there, that you use one purebred animal of our own. In crossbred, we are using the animal which is male, which is not from Indian origin or our native origin, right? But in graded up, we use the animals or males of a purebred of our native germplasm, genetic resource, and we grade those which we cannot classify into one group or population. Suppose there is an animal and we say to Dr. Rana that, Dr. Rana, tell me what is the breed, you are an animal breeder, and he is confusing, he is putting his hand like this, and he says it is non-describable, it is nondescript. So how to breed that? So better to use one purebred animal to upgrade that animal, so that after 2-3 generations, it will become an upgraded one. So, but this also leads to a variation in the germplasm. This was the buffalo which I was telling, that Neeli Ravi. So if in this topmost second photo you see the Neeli Ravi animal with the calf. So this is the pure breeding. 5 markings, the legs, the face, eyes, muzzle, tail, all are white, other body parts are black. But when we made this pure breeding with nondescript, which we don't understand what is actually, so you see a herd which is a nondescript herd. So after say few generations, it will lead to the 3 generations practically, it will become similar to those which are pure bred animals. Coming to the small animals, some problem is there. Next, you do it next. हैक हो गया. You have to stop it. इससे कुछ नहीं है. Yeah. So, skin color, what Dr. Ramachandran sir was telling in the morning, that we have to knock out. It is a difficult thing, but it is possible. It is possible because largely 6 genes are we have identified for coat color in Neeli Ravi, because that is a perfect example that specific regions of the body are having white color. So we tend to see, and when the white and black is there, so the inheritance is very, very tough. There is a tough fight between black and white. Wherever black gets a chance, it will make it a complete black. Wherever the white gets a chance, it will make it a complete white. So these are the contrasts, black and white, good and bad. So that is going on. So the result of, as a result, the geneticist says that we cannot control the inheritance of this type of skin color. Skin color inheritance, though, is governed by, say, 6 or 11 genes altogether, but in a breed, it is difficult to cover. There, I am showing an example where a belt pattern is a well-known pattern in the pigs varieties, that white belt is there. So you see how the belt is varying within a breed. And even after 3-4 generations, these traits come back. It hit backs. You thought that it is white now, pure white or pure black, but after say even after 5th generation of mating, the ancestor which was 5 generations back in the pedigree, same animal will come in the 6th generation. So now, but the technique is there, and that agouti gene and ACIT genes are there. So we have to follow a knockout approach, and that to biallelic knockout. So that leads to the questions of survivability, the aging. So we need to work on that. So recently at NDRI, we tried to knock out the gene which was limiting the muscle growth in buffalo. So we wanted to make more meaty buffalo. So we knocked it out. But it was very wonderful to see that in 1 month, the animal was like 7 or 8 months healthy, but it didn't survive after some time. So people say there are two different school of thoughts, that they say now it has been standardized, everything has been standardized, and it is our technique and this. But if we are using CRISPR-Cas, the very information there which we got it recently to our knowledge, that CRISPR-Cas, if you are using to gene knockout, then you cannot patent it, because CRISPR-Cas is already patented by the company in all the versions. So if you use CRISPR-Cas to knock out, the resultant you will not be able to patent unless the CRISPR-Cas company has granted you permission to do that. So we say Talent technology is there, ZFN technology is there, CRISPR-Cas is there, Cas9 is there. So we need to think on those things. So how on classical level it will go next? Yeah. How the classical level it will go from one generation to generation. So we'll say that we'll use a purebred sire. Sire means a male which has been selected one and is being used largely on the population. Selected sire. So it's called, we say, a sire, and a purebred means belonging to one pure breed. And then these are different grades: 5 by 7 by 8, 1 by 8, and then 3 by 4, 1 by 4. 3 by 4, 1 by 4 means the level of blood inheritance they will acquire from the purebred sire. So after, say, first or second generation, you can have 50-50. You have to go back from this side, right? Purebred sire, and then some graded females which were not known. So after one generation, you will have second generation will be 50-50, 1 by 2, 1 by 2. And practically after 7 generation, it will be 99.9%. That is why you in somewhere it is says, your 7 generations will realize I'll do this thing. In some movie, Allu Arjun will say this dialogue. But so that dialogue has a genetics behind it. So for practical purpose, we restrict up to the third generation in large animals, or because generation interval is quite a big problem. So by the time we say that 75% it is equivalent to the purebred, so we'll stop here and we'll try to maintain at generation 3. So what is happening at the level of homozygosity of the gene? You know that the single locus means the gene which is the house of the gene, the location of the gene is called locus. Single locus biallelic system means 2 alleles are present at locus A, 2 alleles are present A1 and A2. So what is happening to their genotype? So there are 3 genotypes possible for a single locus biallelic system, that is A1, 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 A2, and A2, A2. So A1, A1 are homozygous for both the alleles. A2, A2 is homozygous for the second allele. A1, A2 is a heterozygote. So what happens to the heterozygosity as well as to the homozygosity? That is what it has been shown here. So you have to assess this. Population structure need to be assessed and verified. Once you develop a breed here, then after, say, 2 generations, you need to continuously assess what is the population structure of the breed you have developed. And that is defined in terms of homozygosity as well as heterozygosity. So based on phenotype, we can have breed traits, and another approach could be a molecular approach. So for phenotype, what we do a particular breed and the area in which it is present. So survey is done. What is the population of that particular breed? Then what is the geographical distribution of that particular population or breed? Is the breed particularly associated? Next point is that it is particularly associated with some specific production environment, because the breeds are being developed. You cannot develop a high yielding breed in the field level where the fodder scarcity is there, in Thar area or in some area which is a cold area. So you cannot think of. So whether it has related to some specific production environment or some socio-economic or cultural groups in these areas which are maintaining these breeds, then what is the performance? Their traits are like litter size, fertility, production, yearly weight, weight of wool or weight of milk yield. And does this breed have adapted with those unique traits in this? So for this adaptation and to get the breed to be registration, we need to have a stabilized genetic genomic structure. And earlier, say 15 years back, we were unable to see that. But now there are molecular techniques where we can see whether the genetic architecture of a particular population which you have developed has stabilized over 5, 7 generations or not. So based on breed characteristic, physical traits like color, morphometric like height, withers, body length, tail length, face length, production traits, reproduction traits, some physiology. One breed is there in chicken, which is called as naked neck chicken. In order to survive in hot and humid environment, there are no feathers on the neck, and it is a long neck, which helps in the thermoregulation of that breed. So that is also a basis of identifying that naked neck chicken breed. And based on utility, we say that it is a milk breed, draught breed, wool breed, corporate wool breed. So these are the examples. What are the various physical traits in cattle and buffaloes? Head also, we can say forehead is whether it is concave, convex, or straight. Udder shape. So it is not moving, na? So yeah, next. Utility of the breed. Okay, we'll go from this. So there are other ways other than physical, is that using the microsatellite markers or the candidate gene markers or whole sequencing or other modules. Next. No, yeah. So why there is a need for molecular genetic characterization, that we need to understand the evolutionary relationship between the breeds. Suppose there is a situation where we say we cannot maintain or conserve all shape, so that two nearly known breeds, if That is why they are called genome-wide markers. So entire genome they capture. They are highly genetically highly genetically stable and are having an excellent repeatability with much higher accuracy compared to the microsats. And they allow the fast, high throughput genotyping, effectively distinguish heterozygotes, homozygotes alleles, and codominant alleles also. So the next, the use of SNP for breed characterization. So this is the latest what we do right now, the world is doing. So SNPs are used as a breed identification markers. So if we develop breed, we need to be sure that you have characterized it genetically, phenotypically, and then sufficient number of animals are available in the field or with the farmers, not only with the institute. So it usually involves two stages for SNP characterization. One is detection of breed informative SNPs. So we have now tens of thousands of genes or markers. The maximum marker available bearing chip was developed in Hyderabad only. Your sister institute is there, NIAB, Gauchivali. Huh? So they have created a chip with maximum possible markers, that is 1100,000 markers. But in world over, the 770,000 marker chip is available. So you have a platform like this much of length and that has 770,000 markers, and you can type them whether the allele is present or absent, or if present, in what frequency it is present. Based on that, you can create a reference population. And reference population means common markers you will remove from one particular breed, and you will find those markers which are unique to your breeds, right? When the new breed developed, you need to prune out from those 770, or you can use their chip on 1100,000 markers, and you find out, say, 10,000 markers which are differentiating your new breed. So that breed, those animals which are having those markers will serve as a reference. And from that reference, we will start our breeding program. And from that breeding program and assessing the relationship from the reference, we can say at the time of the birth, we need not to see mature, wait for its maturity or anything. Say if 1 week, you can think of whether the breed will be good, high fertile, low fertile, for our use, our trait of interest is being expressed or not, something, or what we have knocked in, whether they are working or not. So several statistical methods are available. These are the delta method, pairwise FST. It is the between breed variation. FST is the between breed variation marker. That means if the FST is 0, that means your breed is having no difference from the other related breeds. And if FST is 1, you can say that it is, yes, distinct breed. That means within breed variation, among the individuals of the breed is highly variable. But if a breed has to be said, we say that purebred, it should be 0, right? Within breed, there should not be any variation. They should breed alike. That is what is the simplest definition of a breed, which breeds true to the breed alike. And informativeness for the assignment of these markers, their allele frequency, whether they are in linkage disequilibrium. Why disequilibrium? Why not equilibrium? The natural concept is equilibrium. So the nature in a population does to attain an equilibrium. If an equilibrium is attained, all effects of mutation, migration, selection, that goes off. But if you want a marker to go side with the gene of your interest, you need not to have an equilibrium, rather you need to have a disequilibrium, and this disequilibrium leads to the linkage of genes. So that is what is there. And now the breed characterization, since there are large number of markers in large number of individuals, the data size is very high. You can't even visually scroll down. So you can use the various softwares, tools, as well as the artificial machine learning languages, artificial neural network, naive Bayes approach, or support vector machine, or k nearest neighbor. These are the methods which are being used in machine learning to breed assignment. So few more slides, one or two, I think. So those who are feeling drowsy, so happy ending is about to come. So take home message, you should be ready. So breed identification can be done by estimating genomic breed composition. So linear regression we see with the markers and the genotypes and the alleles. And the GBC for a breed is estimated as the ratio of that breed's regression coefficient over the sum of regression. So those who are not from mathematical background, let us understand regression in a bit, that there is a cause and there is effect. So the regression coefficient tells us the relationship between a cause and an effect. Okay. So softwares are there. Harlequin, FStat, GenePop, these are all for microsatellite. And genetic distance, trees, beast, dispen, mega split, cluster analysis, structure, population structure, population admixture, BAPS, instruct, GeneLand. And this is how we assess the molecular variance. Check for the equilibrium. The well-known equilibrium is Hardy-Weinberg equilibrium. Bottleneck analysis. The bottleneck, that what I was telling you in the morning. Sometimes now the population is not sustaining and it has reached due to some bottlenecks. That means the most common alleles are now fading away from the population. The animals bearing the most common alleles are either not bred or they are dying due to some reasons. So the population becomes a risk of going down or to extinction. And do the phylogenetic analysis. Next. So we try next we try to analyze the molecular variance, that is called as AMOVA. One thing is analysis of variance ANOVA, and another thing is analysis of molecular variance. And you can say that the population differentiation is present or not. Another multivariate technique is the PCA, principal component analysis. And this is the beauty of this technique is you can have n you can have thousands and thousands of factors which are affecting the particular trait. So cause and effect need not to be limited to one or two. If there are 10,000 traits which are affecting one particular trait, so you can mix and you can have the assessment. And then Shannon index is also there for relative abundance of genetic information. So here is my last slide. So conclusion is that many evolutionary forces like mutation, selective breeding, adaptation, isolation, and genetic drift have created the diversity. If somebody says from where the diversity has come, so in nature, mutation, when we select to breed, and then isolation from a geographically or different climate, isolation is there. So the population in terms of gene and genotype frequency is drifting from the Hardy-Weinberg equilibrium. So the second is that diverse genetic resources should be characterized for the sake of breed status. You know that this breed is different. Visually, you can say it is different, but we need to do a study so that you can validate that it is a separate breed, and then you can go. And effective monitoring and to establish early warning and response. Suppose there is a threat to biodiversity and it is going to extinct. We don't want that also. So best thing is stitch in time saves 9. So better to maintain it rather than being lost. So molecular characterization and phenotypic characterization help us to prioritize the conservation efforts and decision for taking the animal genetic resources. SNP is the marker of choice and breed specific markers are there. This is with respect to that, and this is especially for on the request of the Dr. Mukesh that we need to go for the steps of breed registration. Only 2 more slides. So Raksa, breed registration is being done to protect our intellectual property right. But in large animals, we are, first of all, we are also registering the strains of the poultry as well as pigs. So there is no doubt you can go on and do it. Side by side, what you have to do, you have to adopt some 10-20 villages where you have to give those what you have developed, so that we can say that it is not the institute which is having a breed. It is the region which is having a breed. So that is one of the criteria. And once you have built for a specific purpose, you need to observe it for say 5 to 8 generations, that these gene has stabilized over the period of generation. So that is there, and this is the basis of breed registration. NBGR is having a registration process. So what it is done that they have the format and everything, you have to fill it and submit the application. The date of application marks your claim, just like in IPR. And what NBGR will do, will preliminary screen the application and either suggest you some changes or will raise a browse on your questions, your claims, right? So they will ask you to supplement something here and there. And next slide. So the examination of breeds by the experts team. So one team comes first from the NBGR or from that species specific institute, like Pig Institute is there, or a combination of these experts. They will visit their region and they will see the breed and your claim, and then population verification in the native breeding tract. Even if we developed a synthetic breed, we need to develop what I said, that you need to give it to the farmers. They should adopt, they should able to breed it and maintain it. They should find it some utility. So that is the biggest target which makes a breed to be nationally accepted as a new breed. Or you can tweak something which is very much relevant, and you can say it is for the experimental purpose, for research purpose, not to be done for the farmers. So we are maintaining that also can be considered, no problem. Then our animal science director, deputy director general, he chairs the meeting of breed registration. And there the experts from NBGR, those who have visited, those who have claimed, the director of the institute who has developed the breed, they discuss and they defend their claim. And if that accession number is granted to that breed, and the gazette notification of that breed is done. So these are the, say, some breed registration committee members. This is the Teni Yo. This is the smallest breed of our native, what we were talking about. So see, there is still whiteness is there. We believe these are purebred animals, but extremities are white in male particularly. So this is the region where it is found: Nagaland, Kohima, Fake, and Dimapur. Main use is for pork, bristle, and manure. I think these are very common usage of all the pig breeds. Average adult body weight is 40 kg. Average litter size is 5 liters. And the age at first parturition is 8.7 months, and dressing percentage is around 67%. Thank you. For targeting. If you have any questions.
Any participant having any questions or any queries regarding the breed characterization and other aspects and concepts about the breed? Yeah, Pridhi, please.
Very good talk, sir. Just small basic query. Sir, which animals, I mean, like inbred or outbred animals are more susceptible resistance to this infectious diseases, sir?
Outbred.
Outbred
More
animals.
is the variation,
And
the more is the chances, if the inbred is there, you can say it will follow the all or none rule. If they are really susceptible or resistant, susceptible you asked, resistance is something different. Inbreeding, if they are resistant to that thing, and that you have a homogeneous line for that, then all animal will be resistant.
Okay.
But susceptibility is varying, it is not all or none. So outcrossing gives us the option to have a wide variation for the susceptibility. Some will have lower susceptibility, mediocre, higher susceptibility, no susceptibility at all. So you can screen among them. So you will have large variation, and it will not wipe off easily. Whereas inbred line, you have to maintain the purebred inbred mating, and that if it is resistant, it will survive the challenge. If it is not, the entire population will be wiped out.
And sir, one more thing, post-vaccination for I mean, outbred or inbred animal. So which is seen more protective, sir? I mean, like either inbred animals or outbred animals again.
See, there is no right answer to this thing, but
Okay.
if you want, inbred animal will be better. Why we make it inbred? Let us ask a question. Why we make an inbred? Inbred means that we are removing the variation.
Yeah, homozygous.
Homozygous for that particular allele or that candidate gene, which is, say, vaccine respondent, some molecular antigen representing cell, depending upon whatever is the virology you can think of. But to test whether it is there, you probably go with the inbred one.
Okay.
And you have to prepare a pure, say, up to 7 to 20 to 24 generations, I would say, in mice is required for making a pure inbred lines. Or you can export it, import it, get it from India, or some laboratories are producing this strain. So I think vaccine के लिए तो available हैं सारे,
Yeah.
है ना?
Thank
All
you,
are
sir.
available.
Thank you.
So it will not be possible to characterize a breed.
No, it is possible at the institute level to reduce a breed, to characterize a breed, to register a breed. But the point is that you have to disseminate to the farmers, either in the form of the semen or the live animals. Just
For research purpose only.
if the purpose is, I told you, research, then the institute can have it. Institute can develop it, maintain it, have it. No problem.
मिनिमम नंबर है सर कोई जो ब्रीड आइडेंटिफिकेशन आफ्टर ब्रीड आइडेंटिफिकेशन एनी मिनिमम नंबर if we develop a breed in an institute then minimum number which we have to maintain
Larger is the better, but in sheep and goat you are working, so at least 1,000 females.
1000 females,
Males minimum 25.
Achha.
Having a sire family of say around 30 progenies each in
sir
the population.
pigs भी same
Pigs में भी लगभग इतने.
और सर अगर एक like we identify one breed in this in the institute or in the surrounding area, so we have to differentiate it from the existing breed also, na?
Existing breed in this region, as well as if you want a national recognition, so from the nation. But if you are able to distinguish within this region, Andhra region or nearby the Hyderabad region, all those districts which are touching the Hyderabad, if you can survey and you can say that this is different from what is being available, say 2 districts in one direction, that makes your claim. Small animals you can. Large animals, by and large, my experience, my focus is on cattle, buffalo. So I feel that large animal doesn't changes from by district to district or by, say, regional. It has to have a regional approach to go. Sometimes the Karnataka, Kerala, and even Tamil Nadu is also involved. So that region specific approach is there in large animal. But small animal will be having a limited distribution. If that breed is having a distribution here and that is limited nowhere else, then also it can be recognized. There's no problem. Sizable number of population. You should assess the population structure. The breed should not be similar to the other known breeds. That means it should not be a variant of the same breed. I told you, na, that some breeds are having within the breeds, the populations are there. So unless that is being associated, so you have vaccine susceptibility, then you can claim it is there. But otherwise, if the genes are same, there is no some variation, and that variation is to the level of broadly, if we say 20%. So we will not consider. If the variation is less than that, then we can say that it is purebred. Some variation, some sire have jumped over the female. You cannot stop it. We don't have that much of controlled breeding. But in lab animals, you can have. There's no problem.
I think we had a wonderful lecture for this foundation, what we call Foundation Day lecture from you. And thank you very much for enlightening us on breed characterization and identifying, starting from the how do we identify, then evaluate and characterize, and then go for this breeding, grading. So the process, I have two quick questions here. Others I'll discuss later. So one is for the food animals, the NBAGR is mainly for the food animals, or is it and for pet animals also? And is it also for the laboratory animals like rat, mice, or anything? That's the one question.
NBGR is definitely not for lab and rat,
Pet.
but it is pet. It has now included dogs.
Uh,
But the mandate given way back in 1980 was farm animal diversity.
So when it
Wild
comes
even
to
it doesn't comes into that.
so when it comes to the registration part,
Yeah.
in laboratory animals, so it's not in the mandate of NBAGR.
But pig and sheep you can.
But that is for food animals. For laboratory animals, it's not coming in the mandate. Now, second question is, within a food animal itself, if a new strain is developed for some purpose, say a pig has been developed for a particular trait, then that strain or line, that also comes under the purview of the NBAGR. So is there any specific nomenclature system for naming them, and how many generations we have to breed them to say that this is a new line or new strain of the animal?
Yeah, that is important, but that varies from the species to species. If you are talking about pigs and poultry, we say 10 generations which are distinct and non-overlapping. If for that generations you have bred it, it is well good enough. It should be in the field. But coming to the nomenclature, it suits you. You say ICMR pig variety 1. That is sufficient.
So maybe you can also, if can share. So we have Indian breeds like Ongol, which has gone to Brazil.
Why I was telling this, we will come to that, because in mice, the lab animals, this is altogether different. They have to have a lab name, strain, population, number, slash, some italics. There is nothing nomenclature like that.
Okay. Now, the second question is some history I would like to, because you have been knowing this and working on this area for ages, I think. So the Ongol, which we had and gone to now Brazil,
Yeah.
Sahiwal that we had gone to the Australia. On the other hand, the HF, which US used to have, now we are claiming our HF. So how does this other country, say whether Brazil or whether India, when they are importing some animal and breeding it, and how do they claim it that it is our breed now? What is the how does they get through this legal process? If you can throw some light on that.
Largely speaking, that what here has gone, it was given by the Maharaja of Bhavnagar 200 years ago. It is very recently, in last 50 years, we have come up with this biodiversity and then IPR issue. And so we were not aware, our scientists were also giving the raw material, genetic material. So they have done. As for now, in large animal or any animal breed, live animal import is very much restricted. So, and export is also restricted. But you can give the DNA, ova, sperm, that we can share, or they can have a MOU sort of thing with some companies, some NGOs in India. They are producing that. So they are selling that as a export. They are allowed to do that. But what Gir and Sahiwal, Australia and Brazil, you are telling, that is that they have taken it from here and they have developed their own. Why I am saying developed their own? There is a big question in ministry also going on. They says it is Gir, but our Gir is GIR and their Gir is GYR. So somebody says it is just the pronunciation, but there is one more breed called Girlando, GIRLANDO, Girlando. So whenever and whatever germplasm from India has been taken abroad, is for one reason only, that is for the thermo resilience, resilience to this ectoparasite and these things. We never imported or exported anything with related to milk. मेरे देश की धरती सोना उगले उगले हीरे मोती, तो ये पूरी खेती पे डिपेंडेंट थी शुरू से आज तक. कभी भी हमने इसको दूध के लिए तो कहा ही नहीं, दूध तो बछड़ा पिएगा, मेल काफ विल ड्रिंक, विल नॉट वीन इट, and we'll have some milk for our household purpose. We never did dairying, right? So our was resilient, hardiness, ability to digest the coarse fibers. These were the traits for which everybody in the world has taken our breed. And on the other hand, we have imported the high yielding, milk yielding variety of the breed. So as I told, that crossbreeding is the only way where you can build up a new germplasm, provided after say so many generations, the genetic structure has stabilized and it has yielded. And that is through systematic breeding only. So they technology, marketing strategy. So they do, Brazil didn't testing, but on the other hand, when America exported imported HF breed. So whatever they say, Brahman breed, it is the Sahiwal of India or Pakistan. Present day Pakistan has been taken from India only. So that has been taken for thermotolerant or adaptation to the tropical environment. So now there is a big question whether that is a breed and whether we need to introduce that breed in India and whether that breed will be much more better than what we have it. We introduced the Holstein, say in 60s, but now we are saying that it cannot be, improvement cannot be made. Heterosis or the benefit of crossing the two breeds goes on by half, decreases by half in the subsequent generation. So first generation, suppose 100 kg has improved. Second generation, 50 kg will be improved. Third generation, only 25, adding to that. 100 has improved, 150 has improved. So 150 has improved, then 175 has improved. And like that, after 10 generation, it will stabilize somewhere. So that they call it as a separate breed. So if that is the breed, and if that breed is producing 40 kg for cattle, that's up. Cattle में हमारे को not only genetic germplasm, but what we are feeding to yield the 40 kg is also important. We don't feed. We leave it to the
नहीं हमारे यहाँ तो ये चाहते हैं कि गाय जाए चर के आए बाहर में शाम को 3 लीटर दूध दे दे
नहीं ये सच्चाई है, लेकिन चाहते थे कि मतलब 15-20 दे दे, तो वो पॉसिबल नहीं है. Our germplasm is sturdy. You can put it to anything. आप उसको कुछ भी नहीं दोगे ना, 2 दिन पानी भी नहीं पिएगी गाय, तब भी वो दूध दे देती है. 2 दिन.
So nice, sir. Thank you very much for this, your excellent talk. Yeah, yes.
We are importing a lot of rats and mouse from the Jackson Laboratory, USA.
Yeah.
And we are actually, when you are importing, so we have to sign an MOU with them saying that we will not breed and sell it to anybody else. But how long we can, what do you say, that we can abide by that law?
Yeah.
So because in every year, so we can produce 4 generations. After how many generations? You say so after 10 generation, it becomes our own.
Yeah, 24 generation.
24 generation. Okay. So then that means 24 generation after that. So legally we are bound to sell. We can sell it.
Yeah. और अब तो मोदी है तो कहते हैं मुमकिन है. Or I to think that sometimes it is good to be an offender. You start selling to India, who will stop? We signed it. Somehow we have to just.
Unless we reserve
that some tweaking need to be done as early as possible and then maintain it for so many generations. Yes. No, it will happen.
They will not allow us to breed. The other way is make them infertile or some kind of gene.
No, it is other way around. Jackson Laboratory must be having their lines, fertile lines, grandparent lines, and they have maintained that. They surely have maintained that variation, because what Dr. Manoj is telling, this will happen. But this doesn't happen to this because the fresh lot comes, so they go back generation and then they introduce something.
Will there be any kind of hindrance there while they are breeding, like thing, or any gene incorporated to not to breed, or any marker that you can identify soon as?
Best of my knowledge, we are procuring Jackson Laboratories for the which are homozygous for some particular trait, and we want to test it. But if you want to breed it, you can do it for any gene. any gene. What only criteria is you have to develop a gene cassette, knockout or knock-in. If you want to add or you remove, do it biallelically, establish that it is there. Then it will come into the germline and there will be no variation in the one generation, and subsequently meeting it for with the parents itself, back crossing with the parents itself, you will attain maximum homozygosity earlier. So sire and progeny, we say father and daughter mating is the maximum in animal. Selfing doesn't exist like plants. So that type of full shift mating, sire-daughter mating, these are the two methods which we can gain maximum homozygosity
It's only a
per generation.
that not breed animals. But otherwise, what is the way to identify whether they have bred it or not? Very difficult.
No, I didn't get you. Sorry.
No. It's only an agreement. It's only an agreement that made between the institutions. Otherwise, it is very difficult to identify whether you have bred my animals or not, isn't it?
You can do it.
You can do
You can do it.
it. That's what I was
I am
telling. It's a
we are
market.
having a very this example I'll tell you with the cattle semen. Cattle we cannot kill in our country, right?
I'm talking about the mice actually.
No, I
Because
give you.
Jackson
I'll
deals
come to
only
that
with
mice.
mice. Yeah.
Come to that mice. So what happened? That West has developed a technology called as sexed semen. So you can have only females. It is a gender-biased semen.
Yes.
So and that is very simple technology. Cell sorting with two nozzles. Separating that and using it and filling it and sealing it. Now for last say 10 years, India is being trying to have an alternative to that. World, I don't know why they are not trying, but world has stopped trying that. It was expected that in 2017, the patent will be gone. But prior to that, in 2015, they take 25 more patents with the same simple technology. By providing that medium, nourishing medium that survives the your sperms, sorted sperms, the sperms which has been hitted by the laser. So they got it and we are unable to do anything. And the moment is that we can fabricate cell sorter, no problem. India is having a mind. But they have the patent that no two lasers can be used for cell sorting. Wherever the two they are using, it is for the semen sorting. So they have patent for that. So China में उन्होंने sue भी किया. तो अब हमारे पास कोई problem, कोई वो नहीं है. The only thing is we get an agreement. We will not use it for research purpose. We will not use it for this. But one thing is always there that government of India says we'll use it for the benefit of the farmer. Rest I don't want to say. Maybe we record over.
Thank you very much, sir, for your talk and taking us through various concepts of breed and breeding. May I request now our director to felicitate the guest of honor with a moment to end a shawl. Please, sir. I would also request our founding director to join the dais. I would also request like to request our director to felicitate our founding director, Dr. P. Suresh, with a memento and shawl. May I request our director to please felicitate our former director, Dr. H.G. Ramachandra, sir. Thank you very much, sir. Please have a seat on the dais. I would like to present the vote of thanks. Thank you very much, sir. On this auspicious occasion of our Foundation Day of the ICMR-National, it's my esteemed privilege to extend the heartfelt vote of thanks, first of all, to our Director General of ICMR, Dr. Rajiv Well, sir, for sparing the time and gracing us on this occasion. We are deeply honored and grateful for his enlightening address. I would like to thank our Honorable Director for his support and encouragement for organizing various events related to the Foundation Day celebrations. We express our sincere gratitude to our guest of honor, Dr. Vikas Vora, sir, for gracing us with your presence today and for sharing your invaluable wisdom and insights with us. Thank you very much, sir. I would like to thank also to Dr. P. Suresh, sir, our founding director and former director, Dr. H.G. Ramachandra, sir, and other dignitaries for accepting our invitation and coming to NARFBR to grace this event. From last few days, my colleagues, Dr. Rana, Dr. Mamta, Dr. Prithvi, Suhas, Bhartidasan, and others have been very busy in organizing various sports and cultural activities. I would like to thank to them all for organizing all the activities. I express sincere gratitude to our administrative officer, Mr. Leela Mukteshwar sir, and Mr. Srinivas ji, our account officer, and Durga Prasad ji, Dr. Ravinder Naik, for the meticulous planning, tireless commitment that has been instrumental in making this event a resounding success. From coordinating logistics to ensuring smooth execution, your professionalism and expertise have been evident every step of the way. Last but not least, I would like to thank to all the participants and to our photographer, Mr. Murli Krishna ji, for covering various events during the celebration, despite the scorching heat outside in various sports activities. Thank you all. Thank you very much, sir. Thank you. Thank you, sir.
सर्वस्याप्त्यै सर्वस्य जित्यै सर्वमेव तेनाप्नोति सर्वं जयते. This institution should go sarvasyapte. It should be everywhere. It should live everywhere. Sarvasya jityai means it will live everywhere. Sarvameva tenapnoti sarvam jayate. Everywhere it should be a victorious, this one, without hearing this.
Thank you all. Now I would like to request to continue the program as per the schedule of the sports activities to all. Thank you.